All right, we covered DNA replication and the proteins that are involved in that. Hopefully, I posted the link for the animation, the Harvard animation of DNA replication for you to look at, sort of so you can see it in two-dimensional as it's going. Hopefully you check that out, and it help explain sort of a lot of the protein players and what they do, and also shows you the trombone model, okay, how sort of it really happens in the cell. All right, any questions on DNA replication? All right, and what those proteins do. Today we're going to cover sort of what happens when there are mistakes made. All right, because ideally, with the end result of DNA replication, the ideal result of DNA replication, an exact copy. Okay, but we know that mistakes can and do happen. Okay, and DNA polymerase, the enzyme that is responsible for making the DNA template, does make mistakes. Okay, but its main role is replicating the DNA template, but it does have other roles. That it will recognize that it made a mistake and will correct it. Okay, so we have what's called proofreading ability. Okay, and ways to go back and change the base that's not in the right conformation, change it so it is in the right conformation. Okay, so as DNA is being produced, or what we call synthesized, it is also being proofread. They're checking to make sure that it's the right base that's in the right order, the right base pair is formed. A to T, C to G, etc. Okay? And if it's not in the right form, it will go back and proofread and change it and make sure the right one's there. Okay? So we have a couple different ways, or DNA polymerase has a couple different ways to go about correcting mistakes. Okay? One of which is mismatch repair. All right? So when there's a C to a T base pair instead of an A to a T, an enzyme will recognize that mismatch, cut it out, and DNA polymerase will put the right base in. Simple as that. Okay? And so just to give you some type of implications what that is, an inherited defect in one of the enzymes and one of the proteins that recognize the mismatch repair, okay, leads to an increased susceptibility, increased chance of developing colon cancer. All right? So that's why they ask, have any family history of breast cancer, colon cancer, because that increases your chances of developing colon cancer in the future. All right? Because that is an inherited mark. All right? This defect, the last statement up there says this defect can allow accumulations of DNA mutations, in DNA. That's pretty self-explanatory given the function of the protein. Its function is to recognize mismatches and correct them. If that protein's not working that way, mismatches are going to be passed on. Causes mutations in the DNA, causes problems, accumulations of DNA mutations, and so on. Questions on that? So, DNA can be damaged in several different ways. Exposure to UV light, chemicals, physical alterations, right? Playing with stuff in the lab that you play with, ethidium bromide, other things, all the environmental carcinogens that you are exposed to every day, including aspartame, stuff you drink, you name it. DNA is constantly, your cells are constantly being bombarded by environmental and physical toxins. Okay? But we don't show up and all of a sudden every day have a new cancer that's involved, right? your cells have ways of protecting themselves, right? And we're going to go through one called nucleotide excision repair. So we have mismatch repair, which we'll go through in a second, which is going to recognize a problem with base pairing, okay? And then we have nucleotide excision repair, where the nucleotide is cut out because there's a damage to that nucleotide, okay? So mismatch repair, nucleotide excision repair. Okay? So here you see, where's my little pen? Here we go. All right? So here you see a problem with this base pair. Okay? It's not a mismatch repair. It is a mismatch, right? Those bases are not the correct linkage, right? It's not the correct pairing. What do you notice about the structure of DNA in that mismatch? Abby? All right, so there's like a bump in it. There's a problem. Doesn't fit the structure. It doesn't fit the normal diameter of normal base pairing. Okay? So a protein recognizes that. All right? A nuclease. A nuclease is a protein that will cut DNA. Okay? So the first part, the root of the word of nuclease is nuclei, right? That refers to nucleic acids. And then ASE is the suffix that refers to an enzyme. All right? So an enzyme that will cut a nucleic acid. Okay? In this case, DNA. Most nucleases cut DNA, right? But if it's going to be one that cuts RNA, it would be called a ribonuclease. Okay? So this is going to cut the DNA where there's a mismatch. All right? And remove this part. So this part right here is going to be cut off, leaving open 3 to 4 bases. Now, DNA polymerase comes in. What is right here? The dark blue that's right there where that arrow is, what's left off? A free 3' OH, okay, which is absolutely necessary for polymerase to produce new DNA. Okay, so it leaves that open, and DNA polymerase. So there's a perfectly good starting point for DNA polymerase to come in, and it will fill in that gap. Okay, so it will fill it. What is left right here? Right there. What's left? A little gap. What protein comes in and a gap between what? What is that back? A ligase comes in and will merge those two, the phosphate sugar backbone. Right? Get a complete strain. Right? Ligase comes in. So now you have taken out the chunk of DNA that's not as damaged or not right and replaced it with the right stuff, right? Nucleotide excision repair. Okay? Questions on nucleotide excision repair? For mismatch repair, it's much more simple. Okay? So, for mismatch repair, here you have DNA, right? And you have bases. Okay? And now say right here, this is the wrong base. Let me erase this part right. So as DNA polymerase is producing new DNA, it will recognize this fit. Okay? It will recognize that this base pair does not fit with this base pair. Okay? And will remove this base pair away and bring in the right base pair so it matches up. All right? This mismatch repair is something that is ongoing during synthesis. Okay, so as DNA polymerase is producing a new piece of DNA off the template, it is doing this proofreading as it is. Okay? How fast are human or eukaryotic DNA polymerases? 50 per second. All right, about 50 per second. Okay, and prokaryotic polymerases are 500 per second. Okay, we said one of the reasons was for this to happen is that the complexity of the DNA is much more, right? So the second is that the proofreading ability of prokaryotic DNA polymerases is much less than eukaryotic polymerases. Okay, so we call this fidelity. We get, like, scientists call this a measure of fidelity. It's a measure of how accurate the polymerases are in producing these DNA templates, right? How error-prone the DNA polymerases are. And prokaryotic polymerases tend to be more error-prone than eukaryotic polymerases because they don't proofread as well. Austin.
Does that mean prokaryotes replicate faster than eukaryotes?
Yes.
Okay.
They replicate faster, and they tend to increase or harbor more mutations because their proofreading ability of their polymerases is quite less. All right. What does this mean in terms of evolution? So, because of the proofreading ability of the enzymes, mismatches or errors, the error rate, tends to be pretty low, right? Very low. But we know that mutations do occur, so the error rate's not zero. It's not a perfect system. And what's the advantage of it not being a perfect system?
Genetic variation.
Genetic variation. Mutations go on. A mutation that is advantageous gets passed on, except if it was at zero, it wouldn't work, okay? And sequence changes can become permanent, right, and can be passed on to the next generation, right? This is important for genetic variation and then eventually for natural selection, right, and has huge impacts on evolution. All right, so that's problems, mismatch, and nucleotide excision repair. All right, one other problem that DNA replication runs into is that it tends to slow down at the ends. All right, what happens, so let's do it this way. Here we have DNA, okay, and we have 5 prime, 3 prime, 5 prime, 3 prime. Okay? And we know DNA polymerase can only add bases at the 3 prime end, right? So as it runs through, what ends up happening is that it doesn't go all the way to the end. Right? So the ends of chromosomes tend to have an unfinished part. So this part will be replicated, right? So it'll replicate up, and then it will sort of not finish. Okay? Is that a problem? Why?
Unpaired.
So it's unpaired, and then what's going to happen over rounds and rounds and rounds of DNA replication?
Lots of DNA.
It's going to get shorter and shorter and shorter, right? Because over time, you're going to lose the ends of chromosomes. Okay? That's a problem. Because starting, say, at the original zygote that's produced, that's your full length of DNA, over those trillions of replications that occurred, less and less DNA is produced. So eukaryotic cells have developed a way to counteract that. All right? And so ends of chromosomes, which are called telomeres, right? This ending, okay? Telomeres are extended, or the piece of DNA that's been replicated, that's a little shorter, has to be extended, okay? And there's a certain enzyme that will recognize that they're not completely equal and will extend the length of the synthesized DNA based upon the template that's there. All right? And that ends, so these are telomeres. All right? And the enzyme that does the extension is called telomerase. Okay? So it's an enzyme that will extend the length of telomeres. All right? So here what you're looking at is a metaphase spread of chromosomes. So it's a DNA dye, and those are your duplicated chromosomes. And then what's up there in red is for the protein telomerase. And you'll see they are at each end of the chromosomes. So that over time, DNA doesn't decrease in size. Okay? So it's a sort of a quality control mechanism to make sure that the full extension of DNA is replicated in every round. All right? So there's special nucleotide sequences at the end of chromosomes that tend to not have very many genes associated with them, so sort of another fail-safe, if you will. All right? And then the telomeres do not prevent the shortening of DNA molecules, but they do postpone it. And telomerase helps to produce that. Okay? The shortening of telomeres and DNA in the overall length of chromosomes has been associated with aging. Okay? So over time, your telomerase, that enzyme that extends and maintains telomeres, okay, decreases in activity. That has been associated with aging. Okay? Now, what's the flip side of that? What if telomerase was active all the time? So you would counteract aging. That's one way of doing it, right? It's not so much that sort of easy. All right, so telomerase catalyzes the lengthening of telomeres in germ cells and in regular cells. Okay? It's not usually active in most human somatic cells, your body cells, normally, because a cell will, if it shortens DNA, what will end up happening is that that cell would undergo apoptosis or cell death and be replaced by another cell. Okay? But in germ cells, when you're producing sperm and egg, when you're producing more cells that are undergoing mitosis, you need to produce the right size DNA. Okay? In cancer cells, all right, it is inappropriately active, which will extend the length of telomeres and make cancer cells sort of, quote unquote, immortal. They don't age, all right? And that is one way of cancer sort of evading cell death, living longer, and then growing more, all right? And telomerase is also currently a target for chemotherapy. The idea is stop telomerase activity in cancer cells, they will age, and then they'll die off normally, rather than sort of having to poison all your cells in the body. Okay? Questions on telomerase and what it does? Okay. All right, we'll do this and then we'll go on. Okay. We know that chromosomes are composed of what? DNA and protein. Okay. The structure of chromosomes, and we know that, well, how long the native form of DNA is what? What's the native structure of DNA? A double helix. And if I was to take one of your cells and take the DNA in one of your cells and pull it out to be that double-stranded helix, it would be about 6 feet long. All right? And so in every one of your cells, that double-stranded DNA represents about 6 feet, about 2 meters of DNA. But how wide, and all of that has to be packaged into a nucleus. About what's the normal diameter of a nucleus? Right, so it's somewhere about 10 to 50, depending on the size of the cell, 10 to 50 microns, micrometers. Right, so we're talking about, see, 2 meters is equal to 2 times 10 to the 12th microns. No, that's not right. Something like that. Anyways, it's a big difference. Right? 2 meters, 10 microns. Okay? So you have to fit that 6 feet of DNA into that little microscopic nucleus. How does a cell do that? Because of the way that DNA is wrapped and associated with proteins, it's a packaging that occurs. Okay? So, the bacterial, let's compare bacterial chromosomes versus eukaryotic chromosomes. Size-wise, which one's bigger? Eukaryotic, much bigger. Millions of base pairs more than bacteria. Okay? We said that bacterial chromosomes, much more simple. No proteins. There's no need for packaging because they're so small. Okay? So they're not associated with proteins. All right? Eukaryotic chromosomes are associated with a large amount of proteins because of the packaging that they have. All right? So we know that in bacteria, there's there's not a nucleus, it's called a nucleoli, right? That there's an area where the DNA is sort of found. It's not packaged in a certain. But in eukaryotic chromosomes and in eukaryotic cells, DNA has to be packaged a certain way. Okay? So in normal cells that are in interphase, chromosomes are decondensed, okay, into a form of DNA and protein called chromatin. All right? And then chromosomes are an order of packaging beyond chromatin. All right? This is a multi-step process of packaging DNA. And we're going to go through this quickly. It's not that difficult, okay? And chromatin changes based upon the cell cycle when chromosomes have to condense, decondense, etc. So you go from chromosomes to chromatin back and forth as mitosis occurs. All right? So, let's go through how chromosomes and chromatin are packaged. Okay? Here is this line represents a double-stranded, all right, a double-stranded helix of DNA. All right? So this is double-stranded DNA. Okay? There are several steps and orders of structure that DNA goes through in order to package them all of it into a cell. All right? The first is the first complex of proteins that are sort of wrapped around, or that the DNA wraps around. Okay? This is a collection of proteins right? This is called a nucleosome. And a nucleosome is a collection of, well, it's a kind of a doublet, right? It's a collection of 8 proteins. Those 8 proteins are called histones. Okay? So histones those 8 proteins come together. They have 2 copies of each of them, right? So you have H2A, H2B, H3, and H4. Those are the histones. And you have 2 copies each of those histones. Okay? They come together to form a protein complex that actually binds DNA. Okay? And DNA wraps around a histone. Okay? Wraps around the histone complex, forming what's called the nucleosome. So really, a nucleosome is the DNA plus the histone proteins. Okay? So now what occurs is that about 150 base pairs separate each, 150 base pairs of DNA separate each nucleosome. So if I was to go ahead and draw a nucleosome here, 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 and then around it goes, around it goes, around it goes. That's the first order of packaging. Okay? So you have double-stranded, that's one way. Okay? Then the next step, right? So you have this long DNA piece that undergoes double strand, right, into the helix. And then that helix wraps around these nucleosomes, and now I've gone from 6 feet of DNA to somewhere around less than 6 inches. Okay? Now, the next step, and I'll show it to you on here because it's a little clearer to see. Whoop. No. Okay, so here are your histones. All right, they're separate DNA molecules that come together, separate protein molecules that form a complex, and eventually the DNA wraps around it. Okay? All those histones, sort of now they form what's called a beads on a string, and that's what you're looking at right there. All right, so you have nucleosomes, which is the histones wrapped with DNA, and now they form sort of this sort of bead necklace. Okay? Now, those beads arrange themselves in a certain way to sort of clump together. And that's what you see here. Right? So you have one, let's do it in red so you can see you have one there, one there. They sort of arrange themselves in a certain pattern so that they fit and compress into a smaller area. Right? And you can see that this form is called the 30 nanometer fiber. All right? So we've started with a DNA double helix, which was 2 nanometers in diameter, put beads on it, wrapped it around a nucleus, or wrapped it around a histone complex to form nucleosomes. Now that's a 10 nanometers in diameter. And now we take 3 of those nucleosomes and pack them really close to one another, and then in a certain way, and now that is a 30 nanometer fiber. Right? So we've gone order of complexity, of compaction. Right? This is a way of compacting DNA into a nucleus. Okay? Then you have that 30 nanometer fiber forms loops. All right? It loops around, making a 300 nanometer fiber. Further, larger in diameter. right? But getting smaller and smaller in length. Okay? And that is the DNA then is looped together to form the DNA that's found in chromosomes. Okay, so different steps. Double-stranded helix, forming the beads on the string, wrapping around histones, forming nucleosomes to form that 10 nanometer fiber, then looping that 10 nanometer fiber in a certain structure to form a 30 nanometer fiber, and then looping that into a 300 nanometer fiber and into chromosomes. It's a very concerted and directed way to compact DNA. All right? And when we get into gene expression, over how these proteins are removed in order to allow access to DNA for transcription, translation, replication. Okay? The important part is understanding the proteins and that this actually does affect accessibility. All right? So the condensed form of chromatin is called heterochromatin. Okay? It's condensed. It's into this sort of protein structure, the 300 nanometer fiber. It is the DNA sequences are very inaccessible to other things. They're wrapped around proteins and protected. Okay? In some cases, there's a more dispersed form. You have to be able to get to the DNA to make RNA and produce proteins and replicate the DNA, etc. All right? And that is called euchromatin. Euchromatin is less condensed and is more active type of DNA. Heterochromatin tends to be inactive and sort of stored. Okay? Questions on heterochromatin? Okay, and it just makes it largely inaccessible. And we'll go over histone modifications when we talk about DNA transcription. Questions on that? On Monday, we'll go over I'll introduce you to some techniques how DNA is used in the lab. Right? You've done it in lab this week where you did transformation. Okay, we'll go over sort of where you can go from there, and then we'll start gene expression.